Okay, so now we're back with all y'all fine folks. That's how you talk about you. It's you, y'all, and if you're from East Tennessee, yuns, which I grew up in East Tennessee, and I'm like, it's either you if it's one, y'all if it's more than one, or if it's a group, it's yuns. When I first moved to Spring Hill, I went down to the 2 ag teachers, and I'm like, what yuns doing? And they're like, what is yuns? I'm like, lots of y'all. So, everybody got used to hearing the Leah language. My kids at school think I'm a little hillbilly, and they are correct. So the only questions I usually get during the year is, what's oil? Because when I do a lab and I'm like, you need a tablespoon of oil, they're like, we don't know what oil is. I'm like, oil. And so, I've been down here in Spring Hill since 1993, but I still have my upper East Tennessee way of chatting. And so they're doing a little bit of techno. So we're going to start talking about DNA. And so we're looking into the Ag in the Classroom folder to make sure that the lessons that are in there, is Laura, or it says Ag, and the folder that we've shared with you all is entitled Ag in the Classroom Lessons. Let me see if I've added Laura to it, because we're going to take about 5 minutes and make sure that stuff is uploading. We are on the school's Wi-Fi, and of course we are still in summertime, and so our Wi-Fi is probably running a little bit slow uploading lessons. So I'm going to share this with Miss Purtle and see if she can get in it. Share, and I'm going to copy the link one more time, and I'm going to share it with Purtle, so that we can make double sure that you can get into all of our activities. What's your email, Laura? Laura CP. I think I've got, we're trying to do not school email. Is that gmail.com? Got it. And I'm going to do AITC, and I'm going to right click and just send this to her directly and see if she can get in our Google Drive. So go to your Gmail and see if it'll let you in. Has anybody tried to access that link we sent you yet? Casey's on the chat room, so we can see. You might not have, because your internet may not be stable enough to run a Zoom and run, trying to download folders.
I have access to it. The DNA folder is empty.
Okay, so it may still be loading. Let's look at the DNA folder. Now the folders specifically, those are actually from the CD. Let's do a test run. Go down to your actual activities. Let's see. And I am going to drop this particular lab in there. And let's see if we can find it. So let's go to my drive. Let's do this. I'm going to copy these activities. We're going to see if you all can get in the 2 labs that we're about to do. I want to copy those. And I'm going to paste them into the lessons. Sorry for the techno glitch. We're trying to upload them right now. Let's try it from this way. Let's see if we can upload files. We're going to go at this from another way of thinking. Ag in the Classroom Lessons. We can at least get our pure activities on there. Come on. And then, if all else fails, let's do strawberry DNA. So I've got that. classroom lessons. We are going to upload files. And y'all that are like, y'all should have already had this done. You are correct. But this is our very first time doing this, and we are learning right along with you. It's like the first time Laura and I did Zoom. We're like, woo! Can you see a lab called strawberry DNA extraction? It's a PDF. See if you can access that activity through your Google Drive.
Strawberry and other DNA are both in the activity show.
So strawberry DNA is in there at the bottom as PDF. Because that's the one Laura's doing. So if you want to go ahead and pull up that activity, nice. Yes, it's all listed as PDF. I will work on the curriculum CD. If your folders are empty, it's probably because I need to copy them straight from the CD. And since my laptop doesn't have a CD drive, I'm going to have to plug in my external CD drive. And you know where it is when you need it. It's at the house. And so if I can pop Laura's up while she's doing this activity, I'll put the CD in her computer, and she's going to leave her Google Drive up for me, and I'll copy it straight onto her computer, and then we'll see if you can get in those activities. So with that being said, that's what our assistant principal always says before he makes a comment. With that being said, we're going to go ahead and go into an activity called Strawberry DNA.
All right, give me 30 seconds. I'm going to type my email into something, and then we're going to rock and roll.
If you want it, it's right there. Do you want to pull it up?
Nope, we're good. And we'll roll right there. Cool. All righty, guys.
Nice.
So we're going to rock and roll, and we're going to do some strawberry DNA. So I like this one because it's really easy, and I prefer to let Leah, with her anatomy kids, doing the cheek cell stuff. Now, cheek cells are really cool, and we do those when we're looking at plant cells and animal cells. But generally speaking, when we're doing DNA with 9th and 10th graders, I don't like them spitting on each other. That's not really my favorite. And with all the corona, I think I'm going to stick with my strawberry, and she can deal with the upper kids taking their cheek cells out. So what I'm going to do first is I'm going to grab my strawberries. I'm going to put them in a little bag. Actually, before I put them in the bag, I'm going to take off the green top. I'm going to toss those. Now I'm going to stick them in my bag. Zip it on up. And now I'm going to squish these. Now my goal here is to get as much of the juice as I possibly can. But I want to also make sure that I'm not pushing them down on the table, because what that'll do is squish them on the table, it can break apart some of those DNA strands. And so that we can see them, we want to make sure we're doing the best that we can to keep those intact. So I'm going to keep on mushing. Okay. And mushing.
Now, while she's mushing, if you don't have strawberries, I've done this blending wheat germ. You could do this with bananas. Kiwi, whatever you've got that you can see a seed in, squish away. And we don't go buy fresh, delicious strawberries. Whatever I've got that week in the fridge, if I can see a seed in it, I'm squishing it, doing it in this lab. So you can use whatever rotten strawberries you have in your fridge, works like a plan.
All right, so I think we had 2 ag teachers. Would you drop what your pathway is? Are you animal science or are you plant science? Drop that for me real quick. So I got a story to tell you if you're animal science. If not, all right, Casey, what we got? We got animal science people or plant science people?
Animal.
There's an animal.
Okay. All right. They're both animal? Okay. Well, that's all right. So your plant science folks, you can do this one. I think it was Katie. Nick, nice. Okay, so you got some plant science people. So this is probably more along your lines. Oh, Amanda. Oh, it's Amanda Lacey. Hey, Amanda. Yes, frozen is correct. Okay, so the one that what I did, because I enjoyed creeping my students out, it's kind of a jam. But so we would always be castrating animals, or I also went to school with a lot of vets that are now back in the area. So on days that they would do neuters, or after we would castrate, I would just take those testicles back, slice them up into little slices, and then you can also get the DNA out of those testicles as well. So I know my 7th and 8th grade science people were just like, oh, what is she talking about? but it did. And it works. It's great. There's lots of DNA in those. Okay, so now I've got it mushed up real nice and it smells really good. And I've got what I call my magic DNA extracting solution. Okay, now for you, this is really simple. This is just water, Blue Dawn dish detergent, and maybe Leah can correct me, but I really like Blue Dawn dish detergent. For whatever reason, it just works better than the orange. It works better than the green. So I always rock and roll with the blue. And then you also put salt in there. If I'm being 100% honest with you right now, I don't remember what the ratios are and how many tablespoons.
I usually just
like you're in the kitchen,
water,
dump.
Yeah.
That's how that one works.
I'm a dumper.
So take some of that and pour your DNA extraction solution into here. And the only thing that's going to do, you use clear soap. Well, hey, if it works for you, use what you got. And I'm gonna mush it around a little bit more, and this just helps to give it a more liquid feel. And so then I'm gonna take cheesecloth. Now, I know Leah uses coffee filters instead of cheesecloth.
I use cheesecloth at school.
Okay, she uses cheesecloth at school. You can find this in like the cooking section at Walmart. So over there where they have all the pots and the pans and stuff. Or if you go to, we have Food Lion. This is over there with like the cake mixes and stuff. I don't know why it's there, but that's where it is. That's where I get it. Okay. And because I'm at school, what I have is a beaker. I'm going to put me a piece of cheesecloth on top, like a niece. And I'm just making myself a little colander.
Okay, so I'm going to flip this. Give me a second.
All right, all right, all right.
Let me flip.
You got it?
Remind me how we flip it.
We flip it like it is. Oop, hold on. Like it is. Like it is. There we go.
All right.
All right. So what I've done is made myself a little colander. And I'm going to very easily hold on to this and get as much of this juice out of this strawberry pulp as I can. That's my goal, is to get juice, not pulp.
If you don't have beakers, use a plastic cup, put a rubber band around it, rock and roll.
Same thing, works just like a charm. Okay, I'm gonna toss this in my makeshift trash can. And once you get here, I find it easier to try to strain my juice out and give my cheesecloth a little twist. Okay, because remember, my end goal is juice. All right. It's going to be bubbling up, and that's just some of that Dawn dish detergent we got in there.
Of course, if kids get wild and squish that strawberries out, it doesn't matter.
It'll work.
Works like a charm every time.
All right. And then I'm going to get my baggie back and put my strawberry in there. That way I don't have as much garbage to throw away. But I've got this nice little sink, so I'm going to toss it right there. All right. Okay, so now I have my juice. And I know this adds an extra step, but I like to use a big open area so that it doesn't make as much of a mess when I'm trying to strain it out. But I feel like it's easier, and I could be wrong, to use a smaller beaker. And I actually prefer test tubes, but I decided to use this 50 ml beaker because I thought it might show up better on camera. So take this, and you're going to do equal parts of everything. So I've got about 10 mils of strawberry juice. And I'm going to come back on top, and I'm going to add my DNA extraction. And again, equal parts. So I'm going to move it up to the 20 line. Now I'm going to go get us that hot commodity.
We've done workshops this summer, could not find any alcohol. Now, if you see this lab on the USDA's, there was a USDA version of it, USDA, I don't remember where we got it, but it's on something we've got. And it called for, it said frozen Everclear works great. I would not suggest that. I don't even suggest using ethyl alcohol because you have to buy it. Just get Red Label. Well, you know, this is Walmart. Red Label alcohol. Now, this year with coronavirus, we couldn't even find Red Label, and we just used room temperature regular alcohol. It still worked great.
My personal favorite to use is denatured alcohol that you find in in the paint section at Walmart, and I like it just because I don't have to keep it cold. Now, we have a freezer in our prep room, since Leah and I are back to back, and we have a prep room in the middle. We have a
That's our prep room.
Yeah, you're looking straight into her classroom. So we have a freezer, but if I'm on the road doing ag in the classroom workshops, I use denatured alcohol because it doesn't have to stay cold. All right, so again, I'm going to use equal parts. So I'm going to move this one on up to the 30, but I'm making a mess, so it might be a little bit more than 30.
Good thing about alcohol is they squirt it on their hands, it doesn't bother them. They squirt it on the table, it's going to dry. They squirt it on their clothes, it's going to dry.
And with all the Corona, it's going to kill the Rona.
So it's actually easy to use.
Yes. So what you're going to start to see is it's going to start to bubble up here at the top.
I'm trying to hold it up for you. Okay. See where you've got this little layer of DNA material?
That's right.
Don't let it stir it.
That's right. So we're going to sit here and we're not going to wait forever, but we're going to wait for it to bubble up and form what looks like a little cloud.
I call it a wad of snot or a loogie because high school boys, they can relate.
That's right. Don't know if it's ready. Let's poke it. Okay, here it goes. All right. So if you can see it, and I'm going to throw my toothpick on the ground. I'm going to pull it up, and that's me a little strand of DNA. See if I pick up a bigger one.
Yeah, they can actually do a swirl. Just get them a little swirl like Laura's doing. And you got a huge strand of DNA.
There it is.
Yeah. Now, the first thing kids are going to say, can I see this under the microscope? DNA is, the nucleic acids are one of the 4 macromolecules. We're not Vanderbilt. We don't have an EM. So, they're not going to be able to see this under the microscope. It's still pretty cool.
Yeah, it's pretty cool. But this is also where we can start talking about other parts in biology or in our life science classes about having to look at DNA. So with DNA, this is how we can create GMOs. We don't create GMOs with anything that we don't already have. All right, I'm going to give Leah a second. She's going to put the camera up on the stand again, and then I'll be right back with you.
All right, let's unflip.
All right.
Oops.
Oh, there's a really good long one.
Oh, nice. Yeah. If you've got little plastic buttercups, because we're going to do buttercups today too, let them have their little strand of DNA.
Take it with them. Okay, so like I was saying, with DNA, that's how we start to create these GMOs. There's nothing scary. There's no chemicals used. But as we talked about earlier with our apple, we're looking at only being able to use this part of our world to be able to create food. All right, so drop it down in the link or in the comments. How many GMOs do you think there are currently? Let's see. What you got? How many GMOs do you think there are currently? Casey, am I getting any guesses over there? Do you know? Hundreds. Okay, so I've got hundreds as a guess. Any other guesses? Thank you for throwing that out there, Ann. Sticking with us. GMS. She meant GMOs, I think is what she meant. Okay, so our winner chicken dinner is going to be Brooke and Lori. And I'm not seeing any other correct answers, but if you said 9, you're correct. We currently have 9 patented GMOs. They're working on the 10th right now. So GMOs are not as prevalent as everybody thinks. Everybody sees the little butterfly on their food, and we realize that marketing is taking over and we're buying marketing. Well, like I said, there's only 9. They're about to patent the 10th. So let's talk about the 10th one that they're trying to patent right now. Does anybody know what that one is? Drop it in the comments if you know what that one is.
Actually, I found a website. Every one of them is. Hold on, I'll see if I can.
And drop that on there. All right, so I don't have any guesses on the 10th one right now. But right now, they're working on trying to patent the banana. Does anybody know how many species of bananas we have? I'm going to go ahead and give you this one so we're not stuck looking at the comments. But right now, there's only 1 species of banana in the United States. Does anybody know where that's grown? I'll have you type that one in. What state's that one grown in? The banana. That's it. Good job. So the banana is grown in Hawaii. And right now, they're experiencing a blight. If you don't know what a blight is, think of it being kind of like a fungus. Has anyone here ever seen Moana? Anybody with little kids or grandkids that love Moana? Yes. Okay. So I'm not alone in my pain. But do you know that part where the blight or the black fungus looking thing starts to take over the island and the coconuts start to rot? Okay, well, that's what they're experiencing in Hawaii, is this blight that's taken over the bananas. So what they did was they found a way to alter DNA to be what we call resistant. So if you're resistant to something, there, whatever you want.
C or B?
B.
B it is.
Okay. Sorry, we're trying to order pizza. Okay, so so blight takes over, everything turns black. DNA, sorry, I'm trying to catch up in my mind. Okay, so what they do is they try to alter DNA to be resistant to something. So if you're resistant to something, it means that it doesn't take over. For example, I'm not allergic to poison ivy. I can walk through a patch of it, never once break out. So my body is resistant to poison ivy because of something in my DNA. Okay, so what they have done is just like with our DNA in our strawberries, they've taken that out and they're able to alter it to be resistant to the blight. Because if the blight were to take over, we only have this one species, and the banana as we know it would be gone, right? So why is that important? Because personally, I'm not a big banana lover, not a big deal. Well, one of our other GMOs is tomatoes, okay? Does anybody know what tomatoes are genetically modified to do? Looking down at the comments. Okay, so Casey just dropped you a link to that. So tomatoes are modified to be able to have a longer shelf life. So they can sit a tomato down on the shelf, and instead of rotting in just a couple days, you've got more like a week so you can eat it. Okay, I hear you. Big whoop, it's not gonna rot. I can use it faster. Well, what about our third world countries that need us to help feed them? How do we get food to them in a timely enough manner that they have enough time to eat good food? That's how GMOs come in to help us, so that we can look at ways to feed people across our oceans. All right, but what about back here at home? Okay, another one of our 9 already patented GMOs is corn. And corn is resistant to insects. Okay, so we have insects that come in and they eat our corn. Well, that depletes our crops. How are we going to feed people? Well, in that case, most farmers have to go back behind and spray a pesticide. And a pesticide is what helps to kill those kill those insects or pests that eat corn. Okay? So by altering the DNA of corn, we're then able to cut down on the fuel we use in our tractor, the cost of the pesticide that we spray, the amount of pesticides that we spray, because a lot of consumers don't want their food sprayed with it. So that also alleviates that issue. So while we're here with our apple, right here, we've got to figure out ways to feed the world. We've got to figure out ways to get food to people that don't have it. And we've also got to figure out ways to cut down on our natural resources, because fuel for tractors and our cars isn't going to be here forever. Okay? So that's one reason why using GMOs is really important.
Okay, Laura, you'll scoot out of the screen for just a second.
All right, I'm gonna
see how to unmute your video. It says blank. Where's that at? Blank. I'll see.
All right, I'm gonna pull this down.
Okay, Casey, I just dropped you all a link to this actual site that I found. This is from an organization called Crop Life International. And when you're teaching DNA, you know, you just don't have to teach DNA as you go. Laura's given us a lot of great information on GMOs, And if you look at this particular website, I'm going to step out of the screen for just a second. I'm going to talk really loud, because I am the loudest human being on the earth. It actually goes over which GMA crops are here, tells you what year they were genetically modified and put on the market and why. And so a lot of people think, well, GMOs are terrible. And the marketing ploy of the world now is they'll have milk on there, they'll be like, non-GMO. Well, there is no GMO milk. That's just a marketing technique to make people spend more money because they say no GMOs, and people think, well, everything's genetically modified. Well, these particular crops that it gives you right here, it tells you exactly why they are genetically modified. Now, I'm not a big sugar beet producer, but look right here at, say, soybeans. If you click soybeans, I'm trying to click, it'll tell you exactly why they are modified the way they are, and it doesn't want to go in there. But if you'll look at this little slide that it gives you right below it, it tells you exactly what that genetic modification is. Because when people think genetically modified, they freak out like they've made some sort of mutant crop. But if you look at, say, field corn, which is one of the earlier genetically modified crops, it gives you specific byproducts that are made from that corn and tells you exactly what it has been genetically modified to do. So right here under insect resistance, well, they put an insect resistant gene in there, which they probably inserted using a plasmid from inserted a piece of DNA probably grown in a bacterial culture, using a vector to make a plasmid, which is a circular piece of DNA that's recombinant or recombined. And you can see that there's specific reasons. They've made it insect resistant, certain insect resistance, herbicide tolerance, and drought tolerance. The reason they've done that herbicide tolerance is it's probably what's called Roundup Ready, which means that farmers can spray Roundup on their field, which will kill weeds, but not kill the corn. Because corn is a plant. You think if it kills one plant, it's going to kill another. Yes, it's in such a small amount that it's not going to be detectable if you go boil an ear of corn. And if you look in the Ag CD, in the 6th through 8th grade curriculum, there's an activity called poison peanut butter that actually mathematically goes through the calculations of pesticides in peanut butter. And it's like 0.0000000000001. I made up all those zeros. But it actually tells you why. Canola, herbicide tolerance, insect resistance, herbicide tolerance, which for people on the earth would say, why do I need it to be insect resistant? Insects don't kill it. Well, insects may actually obliterate a crop if people are in a location maybe where they are growing that crop and don't have access to herbicides or pesticides. And farmers aren't just mowing that stuff out. It is extremely expensive to apply any of those. They want to apply them only on the parts of the field that are necessary. That's why they actually survey to see when it becomes an economic threshold level. They only spray it where it's needed, and they spray a small amount as they can because that stuff is money going out of their pocket. But if you look down here at the papaya, it's disease resistant. If it wasn't for a GMO situation in those papaya plants, they may have obliterated the entire papaya population on, say, a whole island. But then there's islands that refuse to allow them to have genetically modified papaya. And you never know if, that may actually be obliterated from the genome of our plants on the earth. And so always get your kids to research. If you're a science teacher, you know that the science standards were reevaluated a couple years ago. Now, I was one of the trainers that trained on the new state science standards. There are 8 science and engineering practices that your kids are going to be using. If you're not a science teacher, still try to relate these 8 science and engineering practices, which you could easily see on the Department of Education's website. But one of those science and engineering practices that you should be integrating into your lessons is called argue from evidence. And you think, argue from evidence? Why would my kids want to argue? This is the way that you can kind of get your kids into a couple of groups and let them look at the data and let them have a scientific, not argument, if you will, but a scientific discussion on, GMOs are good, maybe GMOs are not so good. and they can only use the data that they've sourced from an academic source. You know, I always tell my kids, you can't be getting on ask.com and using ask.com as your data. You need to be in a legitimate scientific publication, website, etc. There's a big difference in using Scientific American, which is a magazine that might talk about a legitimate source, or People magazine. No offense, People magazine, but I'm sure you're not the journalistic, hub of all that is scientific. So be sure that your kids, if you're using those science and engineering practices, they can argue from evidence, they can design an experiment, they can just research, carry out investigations. All of these are the science and engineering practices that you're going to be integrating into your classroom. Which leads us, are you done with strawberry DNA?
I'm good. I'm ready to roll.
We're going to roll right into our next activity, which is actually what you can do to get the students to get their own DNA. Now, this one is on the Google Drive. It's Wind Your Way Around Your Own DNA. I probably called it that. I don't know. I stole it off of somebody's website 100 years ago. If you're a teacher who doesn't steal stuff off websites and forget where you got it, you haven't been teaching long, because we're good at stealing what we've got. This particular activity is kids using their own DNA. I do strawberry and I do this. For this activity, you're going to need Gatorade. I use this, I can't remember what it's called. Let me put my glasses on and see what it says. It's called Glacier Cherry. And the reason I use it is it's the closest thing I can get to clear Gatorade. If you don't have Gatorade, you could use salt water. This is going to be the equivalent of the salt water we used in the strawberry lab. But my kids, I really don't want to make salt water for my lab, because my salt's sitting in the prep room with no telling what's climbing over it in the prep room. Y'all know how it is. And so I use this DNA, and you only need a tiny, tiny, tiny amount. And so what you're going to do is, this little bottle will do an entire class. So you're just going to give them maybe 5 mils, a teaspoon of the Gatorade. Why do we use the Gatorade? Because Gatorade is full of electrolytes. So this, you could say, what's an electrolyte? Well, an electrolyte is a substance that will carry an electric current. Well, if you know anything about chemistry, salt will carry an electric current. And so when kids drink Gatorade, they're replenishing the ions, or the electrolytes in their body, sodiums, potassiums, etc. And so DNA, if you look on the sodium, let's see what we got. It's got 50 milligrams of potassium and 160 milligrams of sodium. So you've got some substantial electrolytes in here. So what they're going to do is they're going to take it and they're going to swish it all around their mouth to get them a nice big old slobbery ball of Gatorade. I about spit it down my shirt. You need to get them to scrape the tongue with their teeth. That way they can get some good DNA off of their tongue too. And what you're left with is a gross pile of high school / middle school student Gatorade slobber. And so what we're going to do now is we're going to go through the same experiment we did with strawberry DNA, except this one is way easier, because all you need now is frozen alcohol and a little bit of soap. So Laura used equivalent parts. I only use like a few drops of soap in this particular lab. And if you look on that Wind Your Way or the strawberry DNA, I've actually given you the reasoning behind using those particular things in that lab. The soap is there to break down the lipid layer around the cell. And so you can do that. And then the salts there, it has to do with the polarity of the DNA molecule. And so now you've got your frozen isopropyl alcohol. I always tell my kids, just dribble it down the side. Don't pour it in there and make it, we're not making a milkshake. And so with these bottles that I get at Walmart, they have this little tiny hole. They can actually just squirt it down the side. Equivalent part of alcohol to the slobbers. Now we're back to the toothpick. I'm just going to use the other end of Laura's. And you just take your toothpick. And I don't know if you can see that on mine, but now you've got my own little DNA right there at the bottom of the toothpick. And I actually, we could buy little plastic tubs and let them have their own DNA in here. Now, I don't mind dissection, blood, etc., but I hate spit and I hate hair. So I always make them use something disposable and throw their little spit cups in the trash can. It grosses me out. So that is wind your way around your own DNA. Simple stuff you probably already have at your house, minus the cherry Gatorade. Could you use orange Gatorade? Yes. Red Gatorade? Yes. You're just going to have DNA that's the color of that particular, color of Gatorade. And I know it's kind of wound on the toothpicks. I'm going to try to pull it down a little bit so you can see. Little strand of DNA. It's hard to see, but that's their own DNA. Those are really good labs to do when you're teaching genetics to students. And genetics is one of those standards that we all have. Even if you've got lower elementary kids, that's something they can do as long as you keep the alcohol away from them. I also make my kids wear goggles because, I don't want them squirting alcohol in their eyes. But if you don't have goggles, then you pour the alcohol and then let them see it. The only problem I would have with any class is if I suspected a class was going to turn something up and drink it, that wouldn't be the lab I would do for that class. I would use that as what we call an AP, a dry lab. We have wet labs and dry labs that we do. Wet labs are the students doing the labs. Dry labs is I show them how to do it. And so they can actually see the lab, but they're not doing it on their own. Also, if you're not a big fan of dry labs, always do a dry lab before you do the wet lab with the kid, because we've got a lot of students that they would be more interactive if they felt confident doing a lab. So if you model that lab for them, then they feel confident. They've seen it. They know what's supposed to happen. It doesn't make them feel so nervous, and it doesn't make them feel so awkward, because even high school kids, I teach honors classes. Everybody's like, oh, you're so lucky you teach honors classes. Well, okay. Honors kids, usually one of two things. They absolutely want to be perfect on everything, which is very stressful for them. And so a lot of times they might have anxiety and they don't even want to participate if they know they can't do something absolutely perfect. So let them know that it's okay. This is experimentation, hypothesizing, and using the scientific method. Sometimes there's not a right way and a wrong way. And it's so funny because this toothpick's in this DNA and the DNA is actually hanging down into the liquid. So that's a really good lab. So that's our 2 DNA labs, but we actually have a lab that we're going to show you out of the 6th through 8th grade curriculum. We're going to let you take a 5-minute break, and if you've got the curriculum, we're trying to find the curriculum CD materials, but this one is actually in the Tennessee Farm Bureau's 6th through 8th grade curriculum called Human Traits That Are Inherited. Hey, Casey, do I have that one on the Google Drive? We're going to check it out. I think I loaded that one I think I scanned it on my phone and just loaded that one on there so you wouldn't have to dig through the CD to find it. So give us a second. I'll let you get in your Google Drive and find that one. And then we're going to take a 5-minute break. So we'll take I tell you what, at this point, let's take a good 10-minute break. What time is it? It's 11:45. What do y'all want to do about letting them eat some lunch? We're eating at 11:50. That's, what time is it?
Just over an hour.
Oh, we're an hour away. 5-minute break, my friends.